They process lots of the seafoods from the marine ocean area. The people isolate from the vibrio, and they find this chemical AHL, N-acetylhomoserine lactone. It is contribute to the cell-to-cell communication of these bacteria. And then later on what happened? Becomes heavy and heavy, sick and sick. And they're going to form by what? It's kind of like a matrix. You can say it's a matrix, you can say it's a net system, because there is an extracellular polysaccharides to combine them. They become heavy and heavy, sick and sick. And some of them can be dislodged, but the majority of them will be there. And the biofilm there could stay a couple weeks, couple months, couple years. Now, first thing, why this will be formed? And we talk about the food contact surfaces. I'm processing ground beef, I'm cutting. The bacteria is everywhere. You know the ground beef, they have some generic E. coli or general E. coli, some salmonella, pseudomonas everywhere. Then afterwards, some people didn't clean very well. They just pumped water. Then you see the residue there, then the second day, they're still cutting beef, processing. So what will cause this problem? Basically, they failed SSOP. What's that? Sanitation Standard Operation Procedure. This is a major issue for them to have the biofilm. Now, once this biofilm, the heavy matrix happens, what are they going to do? What's a bad thing for that? It is a highly resistant to sanitizer. So I give you an example. If it's a vegetative cell, which means the bacterial cell grow in broth, we only need 200 parts per million chlorine can kill them. Sometimes even in like 30 seconds we can kill them. But once you have a biofilm, what will happen? If this biofilm formed, even you using 1,000 ppm chlorine, it may be only 1 log reduction, which means you only causing 90% reduction. The other 10% is still there. That could cause a problem. So that's the major tasks, the risks for the biofilm. So once the biofilm formed, the people want to do the research to see the genetics behind it, to see the morphology. What we're using? Scanning electron microscope. We can using laser confocal microscope. We can see the 3D images, because we can see how heavy it is, how thick it is, what the weight is of that. So that's why the function of the SEM, CLSM happens. And I mentioned in my research, we could do using a dye, which is a Levy stain. We could see some of them after antimicrobials, we'll see the red color, which means a dead cell. But majority of that, we see the green cell, it still survive. So what we're going to do, we have to use surfactants sometimes. So when you work in the meat industry, some of you in animal science, nutrition study, and the food science, in the future you could be working for the plants. There is the first job, what you need to do is establish a very strict sanitation standard operation procedure. So usually will be very strict, follow the protocols, including physical removing, followed by antimicrobial rinse, and finally will be drain system. Usually we call it a three-step sanitation procedure, kind of could be successfully remove some of the biofilms. But once it's formed, it will be a catastrophe for the industry because it's very difficult to control. And a lot of the bacteria, they could be dislodged, they could become a contamination resource to everywhere. So then some outbreaks happened. But I tell you one thing is in 2012, in Colorado, in the Jason farms, there's outbreaks happened for the listeria on cantaloupe. And the people go there to do the sanitation, to do their sanitation inspection. And the inspector goes there, we gave them a 95 points, A+ grade, just a week before, but later on, the outbreaks happened. And the people find that they did a horrible job of the sanitation of those utensils and the containers, which is processing the cantaloupes. When you do the harvesting, they have to do the processing. So that's one of the examples. It's very possible most of the foodborne pathogens happening in the outbreaks, there actually is not a vegetative cell like the bacterial cell growing in the broth. Majority of them are biofilms from the environment. Because lots of the people, they failed the sanitation standard procedures. So that's the main reason for that. Okay, so that's why I want to introduce you about what a biofilm looks like. Next topic is a big topic. We will have some sets of the exam questions. We will talk about the culture media. Okay, lots of the things we could talk, so we want to, using Blackboard to draw details to mention. And the several bacterial media we will introduce here. Later on, we will be do the practice in the lab. The concept is a little bit confusing sometimes, but we hope will be explained to you very well. And some practice questions there you could do, and we're also going to explain to you next Tuesday when we do the review session. So, bacteria culture media. Bacteria culturing media. The bacterial culturing media could be categorized into different groups. So the first thing first is easy, based on the chemical. Let me talk about physical first. Physical is easy. First thing is physical. Physical status. Based on the physical status, the bacterial culture medium could categorize into liquid, solid, and semi-solid. And this is easy. Liquid, like a broth. And inside of the broth, we already mentioned, you have peptone, you have triptone, you have salt, you have some sorbing extract, you have a beef extract, and you have water. So usually it's yellow color, like our neutral broth. That's easy, it's a broth. Solid agar, we already mentioned. Solid medium, agar, it could be on the plates or it could be on the slants. We did this in the lab. And the semi-solid, usually containing 0.5% of the agar, this is usually for motility test. And we will do this in the lab later on. Okay, this category is easy, based on physical characteristics. The second one is a little bit tricky. Okay, based on chemical characteristics, we could categorize into two different groups. Number one, we call it a defined media or synthetic media. Defined media or synthetic. complex media. Okay, seems like it's simple, but there's a little bit tricky. First thing, what is defined media? It already says defined. That means every single chemical ingredients are well described. Okay, looks like the definition is simple. I have a question for you. Let's say I have a neutron broth. Okay, I just made up something. Okay, I have a peptone, 10 gram. I have a traptone, 5 gram. I have beef extract, let's say 2 gram. And I have a salt, let's say 5 gram. Other than that, or water. Okay, this is underneath in 1 liter. If on the label of a neutron broth, we see the peptone 10 gram, traptone 5 gram, beef extract 2 gram, sodium chloride 5 gram. Is this a defined media or not? Yes or no? Some of them shake hands. Some of them say yes. Some of them no. Some of them nod. No, it's not. Look at the definition. Every single chemical ingredients are well described. Do you know what inside of traptone? I don't know. What are the chemicals there? What are the chemicals inside of beef extract? No, we don't know about that. So, this is a complex media. So complex media is easy. Other than that, it is a complex media. Because you have traptone, peptone, beef extract, we don't know exactly what inside of the chemicals of that. It's a big matrix. So this is a complex media. Neutron broth is a very good complex media. So what are the examples of a synthetic media, is defined media? Look at on the top. That's a media which is a typical BG11, another one is M9. That is a typical synthetic and defined media. Every single chemical you see, potassium phosphate, magnesium sulfide, calcium chloride, citric acid, ferric ammonium, EDTA, sodium carbon dioxide, all these are well defined. Can you see on the top? How about at the bottom? You have these traptone, peptone, some of those beef extract. We don't know. Let's say this is a pancreatic digest of gelatin, pancreatic digest of casein. We don't know what it is. Casein is a protein. Jelly is a protein and mixture with others. We don't know what's a chemical inside. So that's a complex media. So how we define it is a complex media or a differentiated media? If you see the label of the bottle, or the single chemical, which means the chemical formula is well defined, it is a synthetic. It is a synthetic and defined media. Other than that, it's a complex media. When you see beef extract, peptone, traptone, digested protein, it's a complex media because we don't know exactly what are the single defined chemical inside. Okay? So here gives you a little bit of introduction of what a peptide is, a protein hydrolysis. What is the extract? The beef extract, yeast extract, what's the agar? We know that it's a polysaccharide from seaweed, and most of the microorganisms cannot degrade it. I said we cannot eat it, but bacteria can use it. Okay, so that's some media components. That's something right here. Now we're going to move on to another topic. Based on the function of bacterial media, we could categorize them into support media, enriched media, selective media, and differentiated media. This is the four major categories. And lots we could talk. First of all, what is support media? Grow bacteria, basically 35 degrees Celsius, 24 hours, it could be 10 to the 8 cells per ml. Except for fastidious bacteria. So, very simple. Neutral broth is a support medium. Tropic soil broth, we call it TSB, is a support medium because most of the bacteria will grow there, except the fastidious bacteria. So what are the examples? If I want to grow a Campylobacter jejuni, it cannot grow in neutral broth. Although we say it's very important, but we simply pick a colony, go into a neutral broth, like we did in the lab, like you did on Tuesday. After 48 hours, it won't grow. And we tried a lot because it's a fastidious bacteria. Okay? That's why we say except for fastidious bacteria. So generally, the bacteria will grow. Let's say E. coli, Salmonella, Listeria, or Pseudomonas, they all will be grown. That's for support media. What is enrichment media? Basically, it is support media add certain ingredients will let bacteria grow dramatically. How dramatically? Usually can reach 10 to the 9, 10 to the 10 cells per ml. And a good example is blood agar. And we will talk about that real quick. Okay. What we call enriched media, support media, we add something. Originally can grow 10 to the 8th. It's going to grow dramatically, become 10 to the 9th, 10 to the 10th. That's called enrichment media because we want them to grow dramatically for all the purposes. What's a selective media? Certain ingredients in the medium will favor only certain bacterial growth. So I give you a very simple example. If I have a bacterial agar called TSA, tropic soil agar, add 200 parts per million penicillin. Then what happens? Only penicillin-resistant bacteria will grow. most of the general bacteria will be killed because we have a penicillin. So, only penicillin resistant bacteria will grow. As an example for selected media. Last one, differentiated media. What means differentiated media? The color difference caused by biochemistry reaction, biochemical reaction. Okay? That's the definition. Looks like it's simple. But I want to tell you one thing. It's kind of confusing you when you mix them up. The reason is lots of the bacteria media is not only fit one category. They most likely will fit multiple categories and kind of confusing you. That's the key thing you have to understand very well. So we want to talk about the best thing. We gave you some of the examples. Let's talk about the first agar, blood agar. This we will be using in the lab, and I will show you what is blood agar. Blood agar basically is the neutron agar, like what you did in the lab. When we made it at 70% sheep blood, sorry, 10% sheep blood. But why are we using sheep blood? Because the research showing in 1970s, the sheep blood, the characteristics is same as human blood. So they use 10% sheep blood. Some of them using rabbit blood. You see that, but basically it's 10% sheep blood. Okay? It is an enrichment medium. Because of the 10% blood there, it has hemoglobin and lots of the other heavy nutrients. It will let the bacteria grow dramatically. So it is an enrichment medium. It is also a differentiated medium. Why? Based on whether bacteria could hydrolyze blood cell, we will be categorized into alpha, beta, and gamma hemolytic. And this slide, what I show you, is very important. It will be mentioned several times in this class. If the bacteria will partially hydrolyze or lysis blood cell, will turn some of the green color partially lysis blood cell, it is alpha hemolytic. A good example, Streptococcus pneumoniae. Look at the picture. Okay, Streptococcus pneumoniae. If the bacteria is completely lysis blood cell, will generate a transparent zone. This is called beta hemolysis, the beta hemolytic. Streptococcus pyogenes is a good example. This is the pathogen called strep rot, Streptococcus pyogenes. It's beta hemolytic. If nothing happened, we call it gamma hemolytic. There is a bacteria very famous for that, Enterococcus. We will talk detail maybe following next week. So it is a differentiated medium. Because based on their hemolytic of the blood cell, the color will be changed of the colony on the plates. We could be categorized, become alpha hemolytic, beta hemolytic, and gamma hemolytic. So that's something for blood cells. Very interesting. Then we go back to see the slides. You see this guy there, said chocolate agar. Very interesting. Look at the picture, chocolate agar. But I want to tell you, chocolate agar, there's no chocolate there. It's not like an agar, we put some candy chocolates there, become agar. It is called chocolate-like agar. So what happened, this is a traditional blood agar. Heating at 70 degrees Celsius, then turn brownish color. And it looks like chocolate. That's why we call it a chocolate-like agar. However, people think everybody is smart. They took this out. Then connected. 100 years later, people say chocolate agar. But that name is very confusing. There's no chocolate there. It's not made by chocolate. Okay, lots of people will think so. No. Now, what's the function of the chocolate agar? This used to grow fastidious bacteria. For example, Haemophilus influenza, Neisseria, gonorrhea, and the Campylobacter even grow in a modified chocolate agar with vitamin K, with other ingredients. Of course, in the microaerophilic jar. That's a very picky environments and other things we have to emphasize. So fastidious bacteria is for chocolate agar. That's what the picture looks like. And the picture also showing the blood agar, you see those transparent zone, that's beta hemolytic. It's very important for some bacteria. Okay, that's important. Next one, Macanchi agar. You want to talk about the second one. Macanchi agar.